Hello, and welcome to our Insights lecture with Dr. Peter Adams and Dr. Malene Hansen. We hope you enjoy yourselves today and learn something about the biology of aging. We especially want to thank everyone who gives to Sanford Burnham Prebys. Your philanthropy allows our researchers to pursue new ideas about disease and human biology, leading to findings that help us be healthier for longer. Without all of you, this research can't happen. Let's start real quick with the big news of the day, which of course is the rollout of the COVID-19 vaccine. This is an important step forward in our fight against the pandemic. And while we celebrate the development of the vaccine, we must remember that it could not have happened unless scientists were able to interpret how the disease starts, replicates, and affects the body. Without scientists who understood the underlying biology of the disease, we would not have been able to develop a vaccine against it. And that work of understanding the biology is what happens at basic research centers like Sanford Burnham Prebys and at universities and other institutions all around the country. And that's what we're going to hear about today with aging. When Mick Jagger sang, "What a drag it is getting old," he didn't consider that scientists would take that as a challenge. Doctors Adams and Hansen have and continue to develop a very clear understanding of how we age, how that affects the body, and how we might begin to slow down this process. Joseph Pilates, the inventor of the exercise, said, "We retire too early and we die too young. Our prime of life should be in our 70s and old age should not come until we are almost 100. The research you are going to learn about today is helping us to understand how to make that statement a reality. We're also rolling out a new feature today, an interactive poll. We hope to use this tool in the future to make these events even more interactive. For now, we're testing them out. So here's our first one. Most National Cancer Institute designated cancer centers treat patients, but a very select few are basic science institutions. Sanford Burnham Prebys has been one of those few for 40 years. How many basic science facilities have earned the honor of being designated by the NCI? Sanford Burnham Prebys is one of only 7 NCI designated basic research cancer centers. This honor is a testament to the high quality of science being conducted at our institution. All right, it's time for our second poll question. This time we're playing 2 lies and a truth. On your screen, you see 3 statements about Dr. Malene Hansen. One of them is true. Which one is it? While you're making your selection, let me introduce Malene. Malene is a professor in the Development, Aging, and Regeneration program at Sanford Burnham Prebys. Her lab is advancing our understanding of the genes and processes that affect the rate of aging, many of which are associated with age-related diseases such as cancer and neurodegeneration. Her lab studies the molecular basis of aging with a special focus on the regulation of the cell's ability to recycle its own components, a process called autophagy. Malene is a faculty advisor for postdoctoral training at the Institute and a core director of the Nathan Schock Center for Aging Research in San Diego. She is also an avid beach volleyball player. Please welcome Dr. Malene Hansen.
Well, thank you so much. That's very exciting. I want to say hi to everybody. I also want to thank both Joe and Heather for organizing and inviting Peter and I to talk to you about our research today on the biology of aging. So on my end, I am going to tell you about cellular recycling and its role in aging and disease. And so I'll be telling you about some trends that we're learning from simpler organisms, including our favorite model organism, a little worm called C. elegans. And so we don't yet know if these trends are going to hold true in humans. So I won't promise you that, but we surely have some interesting ideas that holds true both in worms and also in other smaller or simpler organisms like mice and flies, as I will tell you today. All right, so aging is something that we're all way too familiar with, myself included. Here's a picture of me from my first scientific endeavor and a Danish biotech company, Novo Nordisk, and then 30 years later here as a lab head at Sanford Burnham Prebys. Another well-known fact, I'm sure most of you are familiar with, is that aging is the biggest and most common risk factor for a number of diseases that I'm sure you're you've heard about before, including respiratory diseases like pneumonia or COVID-19, which is a respiratory disease. And you probably also heard that older people can be more prone to get this disease as well. So the example from this graph could be that Alzheimer's disease, a neurodegenerative disease, is once a person will hit their 50s, there is an increasing risk of getting this disease. However, what's less known is what actually happens during aging in our cells to the components and the different parts of our cells at the molecular genetic level. And so that's what our lab, along with many other labs, are interested in, understanding these molecular or genetic mechanisms of aging that can help us learn new things about aging that can also be helpful not only to fight one, but many of these age-related diseases. That's our general goal. All right, so back to this cell. So you will know that all of our tissues in our body are composed of cells. Each of these cells over time will meet a number of challenges, right? We have physical, chemical, biological agents that can cause damage to our cells. You would probably know UV light, right, would be a damaging agent, including to the nucleus, something that Peter Adams will speak more about, this particular part of the cell. The good news, though, is that our cells over millions of years have devised and developed different processes that can help repair or counteract some of these problems. And I'm going to tell you about one of these processes, which is the focus of our research, and that's cellular recycling. I'm sure you're familiar with household recycling, which is, we use up a part, we throw it in the trash or in the recycle bin. The recycle truck comes and picks it up and brings it to a recycling plant. Well, our cells figured this out or do something similarly, and that is that over, like in a process that basically would encompass taking or bringing different parts of the cells. There's something called mitochondria. They're a little powerhouse of the cells, but other components of the cells like lipid droplets or either invading bacteria, they can be sequestered into trash bins, which in a cell would look more like bags or like vesicles. And these little vesicles can be transported to other vesicles that are equivalent to these plants, so degradation plants. They're called lysosomes. And in those vesicles, they're kind of like little Pac-Mans or enzymes that can chew up these components. And in that way, generate building blocks that can make new and better components to this cell. So this is a normal process that happens all the time. It's called autophagy for self-digestion. It's a Greek word, self-digestion. And so, like I said, our cells can kind of keep house clean this way by turning over damaged or unused components by means of this process. However, if this process doesn't work as well, you can imagine, for instance, if you run out of your trash bags or you don't build these vesicles very well, whatever is sort of upstream of these damaged molecules or the waste would actually accumulate inside a cell. This is not a good scenario, but it's actually very similar to what happens in our cells over normal aging or over these years of time that goes back. So we normally have young, healthy cells that have components that are well functioning, but over time, they will start to accumulate waste or damaged products or components. So this coincides with this process of autophagy, or this recycling process actually tends to fail over time. And you can see how that can contribute to the cell then accumulating waste and not being able to keep up with the load of trash that needs to be sort of taken out. This is no similar, or actually quite similar to the fact that the same process also has been linked to many of these age-related diseases I already mentioned to you. So for instance, in Alzheimer's disease, a neurodegenerative disease, you also have accumulating waste. These are often proteins that tend to aggregate. And you can imagine, again, if this autophagy or recycling process doesn't work as well, you can have problems with accumulation of waste as well. Big question is, of course, how does this happen? And perhaps more importantly, is there a way that we could reconstitute or maintain autophagy to help keep our cells cleaner for longer, hopefully in this sort of beneficial way that can help promote health. So this is hard to do in humans. We don't study humans sort of experimentally. So the question, of course, would be, how do we study aging experimentally in sort of a direct way? And this is where the little worm comes in, as model organisms have been hugely helpful, at least in getting ideas of how to approach this and learn more important links to that end. So like I said, our favorite model in our lab is a little worm called C. elegans. It's microscopic. This is a picture I took with an iPhone through one of our little microscopes, and you can see the size of them next to one of my hairs. So they're tiny, but the real power for the purpose of our research is that they age over a very short amount of time, so only in a few weeks. Will they undergo at least some changes that we can think of are similar or reminiscent of human aging? They stop moving. You can see their skin gets kind of wrinkled. They actually get more infections like humans also do, like we talked about earlier. So there's at least some similarities, including at the cellular level. So many of these cellular processes have to happen in a worm as well, including autophagy. And the cellular recycling actually turns out to look similar in this case. So in a worm, there are also cells, and these look healthy, just like a human cell in young age. But again, the key here is that over just a few weeks, we can start to see changes of an older cell, just like the human older cell, accumulating waste. And interestingly, that also coincides with the same decline in the ability to do the cellular recycling and keeping the cells cleaner for longer. So that poses, again, of course, an opportunity here to ask also in the worm, albeit at a much faster pace, right? What is going on, and how could we potentially help reconstitute this so we can keep recycling better for longer? So to that end, I just want to highlight another major set of discoveries and achievement that's been made, not only by worms, but a number of small or simpler organisms, and that is there is a number of ways that actually we can make these worms live longer. And so one of them, and I apologize, I know this is right after lunch, but dietary restriction or reducing your food intake, in a restricted but not malfunctioning way. Peter Adams will talk more about this as well. There are genetic alterations or even some pharmacological drugs that can be applied to these animals, including the worms, so that they have a longer health span and also lifespan. So, these are sort of super worms that are now achieving longer and better lives. So we like to study those. And what others have found is really a truly exciting observation, and that is that these long-lived worms, again, we study them over weeks, tend to have healthier cells for longer. So even in an older animals, their cells actually have very little waste, and they don't seem to drop autophagy or lose their ability to recycle over time. So they found a way to somehow boost and maintain this process for longer, maintaining their cells in a better shape. So what we're really interested in is, of course, how do they do that? And so we hope by studying them, we can learn something that's really important for, what is it that's being turned over? What is it they put in these trash bins that seem to be healthy for them and make them be able to stave off these kind of problems that comes with age in terms of waste accumulation. And another side of it is, of course, where in the animal would this happen? Are there certain tissues? Worms have neurons and skin and intestine, just like us. Is there a way to think about different tissues that could be, using this process the same as when would it be important to try and maybe modulate it? So that's the kind of thing we can do in worms, and hopefully learn a lot, new and exciting things that can hopefully ultimately inform us about studying autophagy in a broader sense. So I just want to highlight as a little summary, what we and others have found is that all of these different ways that we can make at least the worms live longer seems to be by, at least in part, engaging autophagy for better cellular recycling. And so this is actually also true in other longevity models. So you can study cellular aging in yeast, right? That's a single cell organism. You can study aging in flies. They live for a couple of months, like a fruit fly. And you can also study it, for instance, in mice. They live more like a couple of years. But common for all of these is that they seem to have a similar relationship with engaging cellular recycling as a way to extend healthy lifespan and health span. Of course, the basic question is this going to apply to humans? And ultimately, this is our goal, obviously, to learn and understand how it applies to healthy aging and disease prevention. I don't have an answer for you quite yet, but I want to highlight that obviously health span to us relates a lot to lifestyle choices. I like this little highlight from the web, right, that shows how there's a path perhaps to health. You would all know many of these ways that we can at least improve health. One of them, important one, being exercise. I speak also as a beach volleyball player here. What's actually really interesting is that autophagy does seem to be induced by exercise, also in humans, but certainly in mice, and that seems to be a very beneficial effort as well. So, all these paths to health, personally, I'm not sure if this path to health necessarily needs to come with longevity, but certainly should come with a better life. That's my goal, at least. So with that, I'm just gonna acknowledge my lab. They're a great bunch of people, although we can't really all work together right now. I want to especially highlight to postdoc Jose and Ify for helping me with these slides. And of course, acknowledge our funding opportunities with happy holidays to everybody. So thank you very much for your attention. I'm happy to take questions.
Thank you, Malene. Quick question for you. I guess sort of a chicken and egg question. Is it that autophagy starts to slow down and so more waste accumulates in the cells, or does so much waste start to accumulate in the cells that it affects autophagy?
Yeah, that's an excellent question. I don't think I have a clear answer to you on that. That's one of the things we're trying to sort of understand, right? Like we can try and do that in a timing way to see if trash sort of accumulates first and you constipate, your system. That's certainly one idea. But ultimately, even not knowing necessarily which ones come first, right? If there's a way to boost the process, that could still be helpful, right? To pick up and try and deal with trash that may come sort of later. There's certainly some examples where you can try and boost later. This is only experiments that have mainly been done in cells, and to some extent also in more simpler organisms, where there seemed to be a capacity to engage, even if, let's say, you normally have lost some of the capacity. So that's very, I think, exciting from a perspective of potentially intervene with that process. But technically, we don't know the answer yet.
Are there drugs, say, that would improve or boost autophagy?
Yeah, so there are drugs that, like I mentioned, can extend lifespan. One of them being rapamycin. Another one, metformin, which I know that Peter will speak about in a second. And common again for them, in fact, I don't think we have any exceptions, is that they also seem to induce autophagy. Now, they may also be doing other things. So in terms of just asking whether inducing autophagy is sufficient to give you, let's say, a lifespan or a health span benefits, I don't think we have fully answered that question. But there are certainly a lot of efforts, ourselves included, to try and find drugs that would modulate autophagy, and a prediction would be perhaps that they could also improve health span and lifespan.
Okay. We have several questions about dietary restriction and intermittent fasting. I know I had a boss who once spent 2 weeks restricting herself to 500 calories a day, and that was not pleasant for her mood or for anybody who had to work with her. So can you elaborate on that a little bit? It's not just simply reducing diet or calories, it's also eating the right things, I would imagine.
Yeah, so first of all, dietary restriction or caloric restriction, it's kind of almost a definition, right? Like, because you can also eat as little as you starve yourself, and there's clearly no benefits for that. So typically, again, from the perspective of all the many types of model organisms, even all the way to monkeys, it's typically around a 30% reduction. I know there are also humans who have voluntarily gone into trials on the calorie restrictions, and I think it's about the same. It's about a 30% dietary intake, like reduction. in terms of what you should eat, there's definitely quality to different types of diet that we can give to the model organisms. In some ways, that's why the field, I guess, have changed a little bit from calling it caloric restriction rather than dietary restriction, right? Because there are types of diet. But in terms of what exactly works best in humans, I'm not the expert to that end. But there are certainly ways that one can then, instead of, let's say, changing different types of diet, you can do, like you said, intermittent fasting, that I know a lot of people adhere to, where you would go for a period of time, actually no different than when we sleep. It's also clear from studies that when we sleep, which is technically a fasting period, autophagy will also turn on as a normal response or recycling response to probably clear up what has sort of accumulated over the daily use, right? So this idea that you can maybe super boost by giving longer periods of intermittent fasting is certainly an idea that's out there. I know it's being formally tested further in humans. As to people I know that are conducting some of those trials, it can be hard to do many of those trials because humans don't always self-report well. So often some of those trials can have their own challenges for making very clear conclusions. I think the trends are there, but in terms of giving an absolute and clear advice to exactly how to do it, I think it's a little unclear, at least to my understanding. Yeah.
We have a lot of questions, and we want to be able to get to Peter. So we're going to save most of these questions for the end, and we'll bring Malene back. Real quick, though, right before we bring Peter on, one last question. Malene, are there diseases that can harm the autophagy? Are there diseases that can slow that down or stop that?
Yeah, so I just had a message in there, a little bullet point in there, but that's exactly the point, that autophagy actually is associated with most of the age-related diseases that I mentioned, that changes risk over time for humans. And there is a lot of evidence to show genetically and other ways that dysregulation of the autophagy process, sometimes more or sometimes less, in certain disease contexts, can be associated with the disease. So that's an extra reason for studying autophagy, not just as a path to perhaps improved health span as a general notion from an aging perspective, but also very specifically towards individual diseases like Alzheimer's disease, for instance. Yeah.
Got it. Thank you very much, Malene. I appreciate it. And we're going to bring on Dr. Peter Adams now. So we're going to do the same thing with Peter, and here are your 3 statements about Dr. Peter Adams, 2 lies and a truth. Peter is a professor in the Tumor Initiation and Maintenance Program in our aforementioned NCI-designated cancer center. Dr. Adams coined the term chromostasis to describe the mechanisms that support the stability of chromatin, the substance of chromosomes, to achieve healthy aging. His team was also the first to describe the epigenetic clock in the mouse, a biochemical test to measure changes in the genome to measure age. Peter is the co-editor-in-chief of the leading aging journal, Aging Cell, and is also a core director of the Nathan Schock Center for Aging Research. And in answer to our poll question, Peter is also an avid cyclist. Please welcome Dr. Peter Adams.
Right, great. Thanks, Joe. Thanks, Malene. So I'm going to talk about, following on a bit from what Malene just talked about, also reiterate some of the themes, but also emphasize, and I've tried to kind of capture this, really the importance of keeping your DNA neat and tidy. Okay, and I think you'll hopefully see what I mean by that. Okay, so one of the first points I want to make is that, as Malene mentioned also, the goal of aging research is to extend health span and not only lifespan. Okay, perhaps extending health span is actually more important. Okay, so if you see this person here, might live a typical lifespan of about 80 years old, but for the last, up to 20 years of her life, she might have, frailty and various different chronic diseases. cancer, Alzheimer's, heart disease, etc. Okay, so not a desirable situation. Now, in principle, lifespan could be extended so that that person lives to be 100 years, but then people often think, well, what's the point if you're now going to have 40 years of these chronic diseases? Okay, we don't really want that. Okay, so but the goal is perhaps to extend lifespan, up to 100 years maybe, but more importantly, I think, to actually decrease the period of frailty and chronic disease at the end of the lifespan. Okay, so you can see that this lady is, has a protracted period of health in middle to later age, and a much more compacted period of frailty and chronic disease. Okay, so this is the goal, and we're pretty sure that this is doable. There's very good evidence that this is doable. For example, we can make mutations in mice. This is a mutation in the PROP1 gene of a mouse, which results in what's known as the Ames dwarf mouse. Okay, so this very cute, very small mouse. Okay, but it just so happens that this mouse lives substantially longer than the regular normal mouse. Okay, so you can see that the normal mouse has a lifespan of about little under 2 years, but this Ames dwarf mouse has a lifespan of nearly 40 months. Okay, so much longer. Okay, and this mouse also has a delayed incidence of cancer and other diseases. Okay, so an extended lifespan and an extended health span. And as Malene mentioned, and I think as many of you know, dietary restriction or calorie restriction is another way to achieve extended lifespan and health span. Okay, so this is a mouse lemur. Okay, this is actually a very small primate. Okay, so pretty close to us, and just as in many other species. Okay, if the diet of this lemur is restricted by about 30% typically, then its lifespan goes from for example 6 to 7 years up to closer to 10 years. Okay, so dietary restriction across many different species and organisms will extend lifespan and health span. And we also know, particularly in humans, there's very good data that exercise can extend lifespan and health span. Okay, and so this is obviously there's reasons why I picked this photo. This is my favorite pastime, but find something that you enjoy and do it, because it's good for you. So really our goal then is to understand how these interventions work, okay, so that we can discover new ones and potentially even drugs that do the same thing. Okay, so this is a picture of Nir Barzilai from Albert Einstein College of Medicine in New York. And Nir has been a leading proponent of what's known as the TAME study, okay, taming aging with metformin. Okay, and the reason for this is it's been noticed that diabetics who are taking metformin to treat their metformin tend to have a lower incidence of chronic diseases of aging, cancer and Alzheimer's, for example, than diabetics who don't take metformin. Okay, so it seems that metformin has the ability to promote healthy aging. Okay, and so Nir is leading the TAME study, which will give metformin to normal, healthy, aged adults and ask whether metformin does indeed promote healthy aging and suppression of disease in elderly humans. And that study is about to start. Metformin isn't the only drug that appears to be able to do this. Okay, so there are other drugs and dietary supplements for which there's also good evidence that they can do similar things. Rapamycin, alpha-ketoglutarate, for example, nicotinamide riboside, and NMN. These various different drugs and dietary supplements can do similar things. Okay, so there's a lot of excitement in the field now about the possibility of testing these molecules in humans. But of course, if we want to find, novel interventions to promote healthy aging and longevity, then we first of all need to know, well, what is the cause of aging? Okay. And so Malene has already introduced this and has told us that aging is essentially caused by the accumulation of damage within molecules, cells, and tissues. Okay, and Malene talked about how autophagy is one process by which the accumulation of that damage can be prevented or even reversed. But what types of damage are we talking about here? Okay, well, in actual fact, there's many different types of damage that cells accumulate as they age. Okay, so one, for example, is mutations within DNA. Okay, so these are changes within the sequence of the DNA, which obviously affects the coding potential of the DNA and ultimately the functions which are coded for by that DNA. Okay, and this depicts many different types of damage which are thought to accumulate with age, the so-called hallmarks of aging. Now, one that we're very interested in my lab are these epigenetic alterations, okay, a particular type of damage that accumulates with age. But what do I mean by epigenetic? Okay, what is epigenetics? Well, obviously, we're all made up of different cells and tissues and organs, okay, and so, for example, the heart and the brain, to take two of them, and as you know, the cells in each of these tissues are very different and doing very, very different jobs. Okay, if you were to look at the cells in the heart versus the cells in the brain down the microscope, then they are very different looking cells and obviously very different tissues. But this is in many ways a paradox, okay, because the DNA which is in each of those cells, okay, it's essentially every cell within your body, okay, whether it's the heart, the brain, the liver, all of those cells essentially have the same DNA sequence, okay, the same DNA code. So how can the heart and the brain be different? Okay, well, it's epigenetics which is responsible for the heart cells and brain cells being different to each other. So how does that happen? Well, in each of your cells, okay, there is approximately 2 meters of DNA. And I think in your body, I think there's literally trillions of cells, okay? So this is a phenomenal amount of DNA within your body, 2 meters in each cell, okay? And that 2 meters of DNA codes for 6 billion of these letters, the GATC, which make up the code of the DNA. Now, all of that is packaged into a nucleus inside of the cell, which is typically a few microns across, okay? So packaged into a very small space. And the way it's packaged is at the simplest level, okay? So that's what I've depicted here, these little beads on a string structures, okay? Each of these beads is called a nucleosome. And this consists of proteins in the middle, around which is wrapped the DNA. Okay, so this is a nucleosome. But the epigenome is modifications of that DNA and of these histone proteins. Okay, and there's many different modifications to the DNA and to the histones. Okay, some of which are depicted here. And it's really these modifications to the DNA and the proteins which alter the function of the DNA between a heart cell and a brain cell, and therefore make the heart and the brain the brain. Okay, so that's epigenetics. But an important thing about epigenetics or the epigenome, as we call it, is that it's not a fixed structure. Okay, it's plastic and it changes over time. Okay, and you can see that here. This is an experiment done by Hiroshi, Zong, and Carolina in my lab. And what we have here are the nuclei of cells, okay, and they are labeled with a fluorescent protein, okay, so you can see the epigenome, it's fluorescent when you shine this light on it. But what you can also do is shine a very bright light on it, and that will actually bleach the fluorescence, okay? In the same way that sunlight can bleach things, we can bleach this fluorescence, okay? And so you'll see what happens here. This half of the cell is going to be bleached, okay? So it'll disappear, okay? So there it's been bleached. And now what happens is that the fluorescence is coming back again, okay? You see that? And this happens, you know, on the order of minutes, okay? And the reason that this fluorescence comes back again is because the fluorescent proteins are moving from the unbleached half of the nucleus into the bleached half, okay? So this shows you how these proteins and the DNA that make up the epigenome are moving around, okay? They're not fixed. And this has important implications for aging, okay? Because if the epigenome can move and change, that means that it does so as we age, okay? And you can see that here. So across the x-axis here, we've got 665 people rank ordered by age. And on this axis, we have levels of specific epigenome modifications, okay, specifically DNA methylation, but that doesn't really matter. And what you can see here is that this pattern is not random, okay? If everything was random, these gains and losses of these epigenome modifications, if everything was random, then you just see a single homogeneous color here, okay? But you can see there's very much a pattern to this, okay? And because this epigenome is changing with a pattern over a lifetime, it means that we can actually look at the levels of these modifications and tell how old somebody is, and actually do that very accurately. Okay, so this is from this is an example of that. Here, you know, DNA has been taken from the blood of a person, and from this pattern here, we've calculated the age, and you can see that the calculated age is essentially exactly the same as the actual age, okay? The age as you know, since that person was born, okay? So this is known as the epigenetic clock. And you can see that there's a very good correlation between the calculated age and the actual age. But what you can also see is that there are some individuals who actually age a little bit faster and some who age a little bit slower. Okay, so this person has a calculated age of 72, but an actual age of only 53. So this person appears to be aging more quickly. And actually, it's been shown that people who age like this are at increased risk of disease and mortality. Okay, so if you have a choice of being this person in red or the slow aging person, you definitely want to be the slow aging person. And so we think that these epigenome changes with age are essentially, you know, a ticking time bomb within us. Okay, so here's a half brain, a healthy brain, and we know that within that brain, the cells are, you know, very highly organized, okay, and communicate with each other in very organized and structured ways. But as we age, as I've told you, okay, the epigenome inside the nuclei of these cells is changing, and that leads to a change in the function and the properties of these cells in such a way that the tissue now becomes very disorganized, okay? Some of those cells may die. Some of them just may change the way that they talk to their neighbors. Some of them may even develop the ability to grow uncontrollably, and that can lead to cancer, okay? But their epigenome changes, and that leads to disorganization and degeneration within the tissue. And this, we think, is, you know, a contributing factor to many degenerative diseases, including, for example, Alzheimer's disease. So what we're doing in my lab is we want to test this hypothesis, okay? We want to test this idea. And the way we're testing this is we're going back to this nucleosome, which I've already shown you. And if we strip some of that away and look a little bit closer, you can see that inside of this nucleosome, there are specific molecular contacts, okay, as illustrated here. And these contacts are responsible for determining the stability of the nucleosome, okay, this fundamental repeating unit of the epigenome. And so what we're doing in the lab is to alter some of these contacts to make nucleosomes more or less stable. And then we can ask, well, does that affect the rate of the ticking of this clock? Does it affect healthy versus unhealthy aging and longevity? And ultimately, what we'd like to do is to identify drugs that can also do this, okay? So drugs which might manipulate nucleosome stability and epigenome stability, and therefore affect healthy aging and longevity. And so at Sanford Burnham Previs, we're very fortunate to have really sophisticated robots which will allow us to screen in a relatively small amount of time, you know, or basically search through hundreds of thousands, even, you know, up to millions of molecules for the molecules which might have the properties that we're looking for. And not only do we have the robots, but we have a fantastic team of collaborators at SBP who can, you know, who have the expertise to do this kind of stuff. Okay, so we're working with Michael, Suzanne, and Thomas on these projects. And as I say, the goal is to perhaps find drugs which can promote healthy aging and longevity. And so, in a nutshell, what we would like to do is figure out how to take exercise and calorie restriction and put it into a pill, okay, which, as I say, can perhaps promote healthy aging. But at the same time, you know, I don't want to be the person to say that, you know, this is I certainly don't aspire to for example, take a pill as opposed to, you know, eat healthily or take proper exercise. Okay, I think that there would be many negatives of that for society. Okay, so I think a conversation that society should also have is, you know, who would get such pills and you know, when would they be administered them. Okay, but that's kind of a separate conversation and something to think about. So finally, you know, same as Malene, this is how we meet in the lab these days. Okay, we do, like everybody, okay, we do a lot of stuff by Zoom. This is the current lab of, you know, really fantastically talented and you know, great scientists who it's a real pleasure to have the opportunity to work with. Okay, and without them, we wouldn't get very far. Okay, and so with that, I can also take questions if there are any. Thanks.
Peter, we have lots of questions. First, I want to thank you. I always learn so much from when you speak. Thank you for talking nerdy to us. So we have more questions than we have ever had. I just want to say that we're probably not going to be able to get to them all, but I do want to assure everybody that we are collecting all of the questions and we will present them to our speakers afterwards, and you will be able to get email answers back. So if we don't get to your question, that doesn't mean we're not going to be able to answer it at all.
Yeah, very happy to do that.
Peter, a couple of questions here. One about, like, in terms of epigenetics, how much of what you're talking about can we affect with diet and exercise, and how much of it is just inherited from our ancestors, our parents, or whatever?
That's a good question. I mean, generally it's thought that in terms of longevity, you know, about 25 to 30% of longevity is inherited. Okay, but I think that's, you know, primarily inherited through DNA, through genetics, not epigenetics. Okay, so the other 75% of longevity is determined by lifestyle choices. Okay, diet, exercise, smoking or not smoking, that kind of stuff. Okay, that affects 75% of longevity. And a lot of that would go through what we call epigenetics, okay, in the sense that these lifestyle influences have the ability to alter the epigenome, okay. They're less able to alter the DNA sequence, okay. So a lot of those lifestyle influences do go through epigenetics. So I would say then that, yeah, epigenetics is a big determinant of health span and lifespan. Inherited epigenetics, okay, I mean, that's a more, not quite sure how much of what number I would put on that. I mean, there are examples, for example, Rolf Bodmer here at SBP works on how a high-fat diet in maternal or paternal flies can influence the functioning of the heart in their offspring. Okay, and Rolf is working on epigenetic mechanisms by which that happens. Okay, so there are kind of epigenetic inherited mechanisms as well that affect health and longevity, but I'm not quite, wouldn't want to put a number on that, but there's certainly examples of it.
So I may then be asking a question that you cannot answer in that way. Can we quantify which of these lifestyle changes are more or less beneficial to increasing healthy lifespan? Like if I could only choose diet or exercise, is there one is one better than the other?
I guess that's a difficult thing to answer, because then it gets into, well, you know, how good or bad is the diet, and how much or how little exercise. Okay, so I don't think you can really say, you know, diet versus exercise, because neither of those on its own is you know, black and white. Okay, there's gradations of both of them. Okay, so no, I can't really answer that. I mean, personally, I think that you know, taking some exercise and doing what you can with your diet, and, you know, doing other sensible things like getting, you know, lots of sleep, for example, all of these contribute, and, you know, try to do all of them to the extent that you can.
You mentioned metformin, and we are getting a lot of questions about metformin, including, should we all just be taking it regardless of whether we have diabetes or not? What are some of the side effects of it? Does it limit the benefits of other things like exercise or dietary restriction? Perhaps you could talk a little bit more about that. Yeah,
so I mean, whether or not we should all be taking it. Okay, I mean, I guess that's what the TAME study is meant to determine. Okay, I mean, if the TAME study finds that there is a benefit for health and longevity of, you know, normal, healthy, elderly people without diabetes, and that the benefits offset the detriments, then that will be a case for taking metformin. Okay, so I don't really want to say that. I mean, I think that the TAME study needs to go forward. There is some evidence, though, that, for example, I think that metformin can actually offset the benefits of exercise. Okay, because part of the way that metformin works is by manipulating mitochondria function. Okay, and obviously exercise also works in large part by stimulating mitochondria function. Okay, so there is some evidence that metformin can prevent the benefits of exercise. Okay, which is obviously an important consideration, and not something that the TAME study is actually going to address as far as I know. So, you know, certainly we need to know more. I, for example, you know, as someone who, you know, does, you know, I do a lot of cycling, I enjoy it, I want to, you know, get the most out of it. I don't want to, I personally wouldn't choose to take metformin, because I don't want to, you know, impair the benefits that I might be getting from cycling. So I wouldn't take it. And to be honest with you, there are a lot of these supplements around now that a lot of people are are starting to take. I choose not to take any of them, okay? Partly because, in large part because, you know, a lot of these things, a lot of biology is, you know, very dose responsive, okay? You know, you can get different effects depending on whether you have a low dose or a high dose of something. And so I don't think we really know for most of these things until we've done things like the TAME study. We don't really know, well, you know, what are the proper doses? Okay, so if I was to start taking metformin or rapamycin or NMN, I wouldn't know what dose to give myself, which I know is going to be beneficial and perhaps not detrimental. And I think that's a concern with a lot of these supplements.
Okay. We're going to bring Malene, Dr. Hansen back, and we got a couple of questions about telomeres and whether telomeres or epigenetics are more accurate at calculating age, and are they related to the genes in the dwarf mouse. Can you explain very, very briefly what telomeres are and what effect they may have on this?
So telomeres are the ends of chromosomes. Okay, so chromosome is obviously a linear piece of DNA, and so it has ends to it, obviously, and those ends are called the telomeres. Okay, and they have, you know, a bunch of important functions in terms of, you know, chromosome function. Those telomeres get shorter as we age. Okay, and sorry, I've lost. What was the question about telomeres again?
It was, are they better than epigenetics, sort of at determining that?
So yeah, it's very clear that actually telomeres, if you look at telomere length with age, okay, there is across a population, a large population, there is a broad correlation in the sense that, you know, telomeres do shorten as we get older, okay, but there's a huge amount of inter-individual variation, okay, so it's very difficult. You basically cannot measure the telomeres of an individual and know how old that person is, okay. In contrast, the epigenetic clock or the DNA methylation clock, or some of the other clocks that are coming out now. I mean, there's metabolic clocks as well, for example. These other clocks, you can take a measurement in a single person and know how old that person is, okay? Because it's not just that it correlates over a population with age, but it's also a precise measure of actual age in the way that telomeres are not. And I don't think there's any dispute about that. In terms of the Ames dwarf mouse, well, this is one of the quirks of mice, okay? This is why mice are not, some of these caveats to some of what we do, okay? Because mice are not the perfect model. Mice have very long telomeres, okay? Much longer than humans, okay? And they also maintain their telomeres with age. So, you know, comparisons between telomeres in mice and humans are not really that helpful. Mouse telomeres are fundamentally quite different.
Okay. Malene, I want to go back to you. There was a question that came up during your talk about taking meds that reduce stressors. And can that help longevity? And I guess I would expand that to, does it help or hurt? You know, what is the effect of that?
Well, first of all, since I've been sitting here watching the question channel, I'm very impressed with all the many questions and how good they are. So it will be a pleasure to address more of them afterwards if we don't get to them here. So to your questions about stressors. So what I also had on the slide is that there is sort of this concept called homeostasis. So think kind of like what doesn't kill you would make you stronger. There is a way, especially of course again in model organisms, that you can give a small stress, like a heat shock, a small heat shock that would then keep you prepared, actually by means probably of epigenetics, like Peter talked about. So your genes are sort of geared, right, and ready to go once you meet another stress that may be stronger and that could be more detrimental to you. So that's a process that we're interested in studying as well. It also seems to boost autophagy in this beneficial manner. So whether there's a way to kind of just get it right, because obviously you can also get too much of a bad thing, and that that wouldn't help you either, right? And there's probably ways to superinduce autophagy that is not helpful either, right? You can overeat your cell, like even by the kind of pictograms that I showed, right? That would also eventually be detrimental. So I think the question about stressors and whether to modulate stress is going to be just as important to do those dose response effects that Peter was talking about directly with the drugs. That said, supposedly there's a lot of benefits going to the sauna, at least by people in Finland. It seemed to improve cardiovascular health, at least, whether that would be similar to giving some of these, beneficial heat shock and warmth, we don't know that. But we like to speculate that it's probably involves some of the same mechanisms. So learning about it, right, and figuring out how we can modulate it in the right way, by the right dose, the right time, could hopefully be helpful to that end. Yeah.
Again, in going through some of the questions, some were asked by folks who chose to remain anonymous. I want to try to get to those since we don't know who to email about them. For both of you, the question was asked, what sort of an effect would chemotherapy have on aging?
Oh.
So I can comment on that because we're doing a little, so it's clear that chemotherapy does accelerate human aging. So this has been noticed. I mean, so a lot of chemotherapies are very toxic to DNA and other molecules in the cell. And so it has, it's been noticed that individuals who get chemotherapy do show certain features of accelerated aging. And that's, that has also been recapitulated in mice and investigated mechanistically. So there is certainly, an effect there. But, if anyone asking the question, I mean, I don't want to cause, kind of undue concern. I mean, I don't think it's not a huge effect, but there is an effect there.
Okay. And then one last question before we finish up. Is there any progress in discovering ways to measure autophagy in real time? Is that?
You mean in humans?
Yes.
I think that's still very early days. For one thing, it's particularly difficult to measure autophagy, and I saw there was a couple of good questions about that as well. Like how do you measure autophagy, including in humans? So there's been some efforts to that. As you can imagine, the easiest cells to get is our blood, because you can take a blood sample and look at the various cells that are in the blood. So there has been some analyses of that. There has been some analyses in mice and sort of vertebrates to that end to look at tissues where you can dissect the tissues, right? But for humans, so far most of the experiments have been done either in blood or in isolated cells to that end. And so the study that comes to mind in terms of thinking about the concepts that we're also trying to put forward is that there are humans that live exceptionally long, like centenarians. Near Barcelona, who Peter Adams mentioned before, is studying a number of these groups. And there's at least a few studies, including from groups at Albert Einstein also, that have shown that autophagy seems to be boosted, at least in these blood cells of the centenarians. So that doesn't prove that they live long because they have better autophagy in their blood cells, but it's certainly consistent with this idea. If there are ways we can find and boost autophagy in a beneficial manner, that could be helpful. But we definitely need better biomarkers and better ways to measure autophagy. That goes for many aspects of aging, actually, to eventually also be able to do something like more like personalized medicine and efforts to that end, which I also believe is an ultimate goal. But there's a lot more work to be done. I should not forget that it's 30 years that the genetics of aging field has really worked on this, right? And it's come a long way. So that's also why we need to train the next generation of researchers, right? Because it may not be in neither mine or Peter Adams's lifespan that we will see all the benefits of this knowledge, but there's definitely a lot to be learned and still focus on for the future. So very exciting times.
Thank you very much. I guess actually I lied. That sits at one final question. Let me ask one more. Is there an outer limit to how long somebody can live healthy based on what we have now? Is it 110 years, 120 years? Is there, what's the number? What are we getting to?
Well, there is one person that is like the most, or at least reported long-lived person. It's a French woman, Chromatin, and she lived to be 122. She didn't seem to suffer many, age-related diseases. So, that's really an interesting situation as well, right? Why would some individuals be able to live much longer than normal? I don't know if she's biobanked anywhere, but if she was, I guess that would be a very interesting person to try and understand better.
Peter, any you want to throw a number out there, or?
Well, I mean, there was a fairly controversial study a couple of years ago which claimed to have looked at the kind of rates of progression of maximum lifespan and did kind of come up with like the 120, 125 figure. But as I say, I think the way they did the analysis was quite controversial. I think a lot of people intuitively kind of, would agree with that. I think objectively, if we accept that, that aging is this accumulation of damage, then I don't think in principle that has to be the limit. And, maybe it could go substantially longer, but I don't know, as I say, it's not really, in my opinion, it's not necessarily what we should be aspiring to. Okay. I think we should be thinking more about, well, how healthily can we get people? Sure. And I would hope that, I would like to see, a time when, perhaps we can all live to 100 years old, and the vast majority of that time we are happy and healthy and productive.
I agree.
Reasonable goal.
Well, thank you both very much. We still have over 35 questions, which we don't have time for, but as we said, we are going to take them and we will forward them on to Peter and Malene, and we will get answers and get back to everybody. Before everybody leaves, we have one more poll question for you, and we would like you to tell us what you'd like to learn about in some of our future events of this type. So you will see you can select more than one. So please do that, and we will take that and we will use that to help develop other lectures in this series. In the meantime, you can also see our future, the ones that we have immediately coming up as we start 2021. Also, if you have other questions that you think about afterwards, or if you'd like to learn more about our research, or even most importantly, if you'd like to contribute to any of our research and support the great work that Malene and Peter and all of our scientists are doing, you may reach out to me, and that is on your screen right now. We want to thank you for attending today. The presentation was recorded. We will send that out to everybody. Please feel free to share that with your friends and family and anybody else who might be interested in it. And we want you to have a wonderful holiday season, and we look forward to seeing you in 2021. Thank you very much.